coming to the synthesis of hemoglobin molecule. So hemoglobin molecule can be synthesized as, we already discussed, hemoglobin molecule need two things for synthesis, a heme portion and a globin portion. So this is, for example, this is a cell. This is a molecule, this is a cell, bone marrow cell. There are special proteins. These special proteins are called transferrins. Transferrins carries two molecules of iron, and these transferrin will bring down this iron. Transferrin is a protein molecule which is bringing down iron component. So this iron and transferrin comes down and it will liberate its iron molecule. Or this iron can be also come down from a stored form of iron. This is stored form of iron. And stored form of iron is called ferritin. This ferritin can also give its iron molecule. Ferritin can also give its iron molecule. So this is story of iron. We have already discussed, let me draw here for you, heme plus globin. Heme is made up of two, iron plus porphyrins. So we have discussed this iron, which is in ferrous state of ferrous state. Now coming to the porphyrin, how this porphyrin is synthesized. In every cell there is a powerhouse of the cell. This powerhouse of the cell is called mitochondria. In this mitochondria, certain substances combine together. Certain substances are succinyl-CoA plus glycine plus vitamin B6, which is a water-soluble vitamin. So these three, succinyl-CoA, glycine and vitamin B6. Succinyl-CoA come from the glycolysis or the Krebs cycle. By the breakdown of sugar molecules, you get succinyl-CoA. Glycine is an amino acid, and vitamin B6 is the vitamin. So there is a consequent steps of reactions which will be discussed soon. So there are 6-7 steps which takes place, and ultimately you get porphyrins. These porphyrins combine together with iron molecule and you get a molecule of heme. You get a molecule of heme. There are ribosomes present. There are ribosomes present. Ribosomes help us in the formation of alpha group, beta group. It is a dimer. Why dimer? Because two, one alpha, one beta. Then one alpha, one beta, as we know that this cannot form a molecule of hemoglobin. So from this dimer molecule, you get a tetramer molecule, two alpha and two beta molecules. Two alpha and two beta molecules combine together with this molecule of heme. Combines together with this molecule of heme, and you get hemoglobin. And you get hemoglobin. So these are all of the reactions, how this synthesis is taking place. This iron will be coming down either with the help of transferrin or already stored form of iron, which is ferritin. Iron will be liberated, iron will be liberated, and there are three very important substrate molecules, succinyl-CoA, glycine and vitamin B6, and there are a lot of steps which we will be discussing later, inshallah. So there will be porphyrin. So porphyrin combined with the molecule of heme, and these are the ribosomes, dimer, a tetramer, and you get a molecule of hemoglobin. Why you want to synthesis, or what is the stimulus of the synthesis of RBC molecule? So whenever there is hypoxia, whenever there is less amount of oxygen supply from the Whenever there is less amount of oxygen supply from the lungs to the tissue, this condition is called hypoxia. Whenever there is hypoxia, the synthesis or the stimuli carrying the stimuli are being transported from one area to another area, and the synthesis of RBC will be enhanced with the help of erythropoietin. Erythropoietin is a hormone which is secreted from kidneys, and this is a stimuli for the synthesis of the molecule of the hemoglobin. If you remember, there was a powerhouse of the cell, so we write a sentence: some great doctors prescribe highly unlimited cancer prescribing pills. Cancer prescribing. So S from this S we get succinyl-CoA, from G glycine. So succinyl-CoA combined with glycine. From D, delta amino levulonic acid. From P, phosphoglycerine. From H, hydroxy methyl benzene. Urophosphinogen, coprophosphinogen, and then protoporphyrin or porphyrins, and then you get. So these are those steps. These are those steps which are needed for the synthesis of the molecule of hemoglobin. From this we come up to know that there are a lot of steps. These steps are: some great doctors prescribe a highly unlimited cancer prescribing pills or heme. So some glycine, succinyl-CoA, delta-amino levulonic acid from prescribing. From this P, phosphoglycerine, hydroxymethylbenzene, urophosphinogen, coprophosphinogen, porphyrins, and then you get a molecule of it. So all of these steps are very necessary for remembering. So please remember this mnemonic. After you remember this mnemonic, you can come up with this thing because it was very difficult to memorize. So remember the mnemonic: some great doctors prescribe a highly specific cancer prescribing pills. So by now, the last thing which we have to discuss, hemoglobin molecule. Hemoglobin molecule were further divided into two, a heme and a globin. Heme was further divided into iron and porphyrins. This globin is made up of amino acids. So there are three very important amino acids which are predominantly present inside human body: leucine, isoleucine, and the last one is histidine. So these are three amino acids which combine together in a relatively higher amount, relatively bigger amount, for the synthesis of this globin molecule. So by now we come up with this, the summary of this hemoglobin molecule. Heme and globin, make sure you memorize everything, make sure you memorize the values, make sure whenever they are synthesized, how they are synthesized, when they are synthesized, and what is the fate of this molecule of hemoglobin inside an RBC. After the 120 days, the destruction will be taking place inside the spleen, and whenever the destruction will be taking place inside the spleen is that's why it's called the graveyard of the RBC. And after the RBC breaking down in the spleen, the molecule of hemoglobin will come out, and the molecule of hemoglobin will split up into two, and that will be the fate we have already discussed. From that, you get unconjugated bilirubin. Liver will be responsible for the conjugation, and from that conjugation you will get a conjugated bilirubin. Why you want to do conjugation? Because you want to excrete that waste product from the body. So this was the everything. Next it will be the different types of molecule of hemoglobin, which we will be discussing inshallah.